observations, write down in your book only that young shoot arises from the node of the stem where there was already leaf. So that portion will grow. Whereas no shoot arises from the stem in which there are no leaves. Clear? So young shoot arises from the node of the stem which has leaves, and whereas no shoot arises from the stem which has no leaves. So it concludes that the presence of a node in stem cutting is needed for the appearance of roots and young shoot. कि जिसमें node होगी, presence of a node is important. So presence of node in stem cutting is required for the growth of roots and young shoot, and the young shoot never arises from the internodal region in stem cuttings. जब आप एक कटिंग लेते हो स्टेम की, उसमें अगर फॉर एग्जांपल देयर इज नो लीव इन बिटवीन, आप एक सिर्फ स्टेम का पोर्शन ले रहे हो, सो देन that no growth will take place from that internodal region. You need to have presence of node is important for the growth of the shoots. Clear? So this is the observation of this activity. Next is one tissue culture is left. So what is tissue culture? We have studied so many methods of vegetative propagation. कितने सारे methods हमने कर लिए, grafting, layering, cutting. So then this is the last method of vegetative propagation only. Okay. So what it is advanced scientific vegetative propagation, it is called. So the methods which we have studied, that are also the methods of vegetative propagation. But if you say that, if anybody asks that which is the advanced method of vegetative propagation, so then you will say tissue culture is the advanced scientific vegetative propagation method, and it is also called micro propagation. It is also called micro propagation. Micro means very small pieces you are taking from the mother plant, and then you are growing the new plants from that small tissues. And how it is done, the process. So what is tissue culture? Tissue culture is a process that involves exposing plant tissue to a specific regimen of nutrients, hormones and light under sterile in vitro conditions to produce many new plants. अब इसमें क्या हो रहा है? For example, you are taking the tissue from the stem. आप stem को scrape करके थोड़ा सा उसमें से tissue का part ले रहे हो, and then you are placing those tissues in a petri dish. If you have shown, I have shown you last time, I think, petri dish. So small, the transparent plate is there, that is called petri dish. In that you are putting those tissues, and we have to provide proper medium to the tissues for the growth. Now what is that medium? वो जो मीडियम हम उसको प्रोवाइड करेंगे टिश्यू को, दैट मीडियम मस्ट कंटेन प्रॉपर न्यूट्रिएंट्स व्हिच आर रिक्वायर्ड फॉर द ग्रोथ, मस्ट कंटेन प्रॉपर हार्मोन्स. कुछ प्लांट हार्मोन्स होते हैं which are required for the growth of the plants. In the last chapter that is deleted from the syllabus, but in control and coordination these all hormones were there. So auxins and cytokinins. Auxins, cytokinins are the hormones. इनको plant hormones बोलते हैं. They are responsible for the growth of the plants. Okay, and they are provided in that medium. वो medium में nutrients and hormones provide किए जाते हैं, and it is done in vitro conditions. In vitro means laboratory conditions. So this method, this procedure is done in the laboratory under sterile conditions. Sterile means free of all infections, free of all microorganisms. So that is a disease-free environment we are providing to that tissues from where the new plants will grow. So we are taking small tissues, we are taking tissues from the mother plant by scraping the stem, and then we are putting those tissues in a petri dish that contains nutrients and hormones, and proper light we are providing, and it is done in the laboratory in sterile conditions. And from those, what will happen initially when you have taken those tissues? So what will happen? Ab cell division ke liye jo nutrients, hormones chahiye, we have provided that. So what will happen? That cells of the tissues will start dividing. वो cells जो हैं, that they will start dividing, and you will get double a number of mass of cells. So जितने tissues में cells थे already, so that will become double in number, and that mass of cells, that group of cells is called callus. It is written in your book also. See the paragraph of tissue culture. Third line. In tissue culture, new plants are grown by removing tissue or separating cells from the growing tip of the plant. The cells are then placed in an artificial medium. So we are providing artificial medium. These tissues are not growing naturally, where they divide rapidly to form a small group of cells called callus. So what is that small group of cells called? That is called callus. Underline this word, Callus. Callus. So this is a mass of cell. When you have taken the tissues, you are growing them in artificial conditions in the laboratory, and we are providing proper nutrients, hormones, and the cells of the tissues, they start dividing, and when they become a mass of cells, so many cells it becomes, that is called callus. Callus. Now this callus is again transferred to another medium. Okay, so it is again transferred. It's transferred to another medium containing hormones for growth and differentiation. See, the plant hormones are different. For the growth, the different is there, and cytokinins are for cell division. Okay, some hormones are for elongation and some hormones are for differentiation. So we don't go to no need to go in the details because it is not required. You just simply write that it is transferred to another medium containing the hormones which are required for the growth and differentiation. Then what will happen? You will see that after few days small saplings will come from those tissues. So जो mass of cells है, जो callus है, उसमें से small plants will come out. On that only, the plantlets are then placed in the soil. So then you can take those small plantlets and you can place it in the soil so that they can grow into mature plants. And using tissue culture, many plants can be grown from one parent in disease free conditions. I told you it is done in the laboratory conditions, in the artificial medium. So there are no chances of diseases because the environment is sterile. It is free from all bacteria. This technique is commonly used for ornamental plants. Ornamental plants are the show plants, which are just normally they don't bear flowers. So ornamental plants are the show plants which are normally you can say reproduced by this method of tissue culture. So I hope it is clear. Tissue culture, you are just taking the tissues from the plant, from the mother plant, and you are growing them under artificial conditions, providing them nutrients, hormones, and proper light. And then these tissues, they divide, the cells of the tissues divide themselves, and they form a mass of cell that is called callus. And from these callus, this callus is then transferred to another medium of hormones which are required for growth and differentiation. And from this callus, after a few days, you will see small plantlets will grow on this callus, and then you can take that small plantlets and then you can place them into the soil and new plant will grow. So this is advanced scientific vegetative propagation method and also called micro propagation. I hope it's clear. So then after that question answers, I hope all of you have done the question answers in your notebook. I have given you the answers of these 5 questions which are there on page number 133. Okay, so then after that, let's start with the sexual reproduction. Okay, so sexual reproduction, what it is. So before we go into the details of sexual reproduction, I want you to know some important terms. Some important terms are there which frequently comes when we talk about sexual reproduction. So that terms should be clear in your mind. So I have taken down some important terms so that it becomes clear to you. Okay. So first is sexual reproduction. Sexual reproduction, what it is the production of offsprings or individuals by the fusion of two gametes to form a diploid zygote which develops into a mature organism. Now you will ask, ma'am, what are these gametes? What do you mean by diploid? What do you mean by zygote? So that's why it is important to know the meaning of all these terms. First of all, just take down the definition that production of offsprings or the individuals by the fusion of two gametes. So we are taking two gametes here. One comes from the male and one comes from the female. Okay. how it produce, how are they formed, that all we will study in the process. But now just take down that what do you mean by this process. It is the fusion of two gametes to form a diploid zygote, and this zygote then develops into embryo, that is fetus, which is there, which then grows into a fully mature baby. So then fertilization, first of all you should know what is fertilization. Fertilization means fusion of two gametes. जब male cell, male gamete and female gamete, they fuse together, they fuse with each other to form a single cell called zygote. So this process of fusion is called fertilization. Okay, you have to learn these definitions. Then haploid, what do you mean by haploid cell? Haploid cell and diploid cell क्या होते हैं? Haploid cells are those which are having single set of chromosomes. I told you that chromosomes are present in the nucleus of the cell. एंड नॉर्मली इफ यू टॉक अबाउट, नॉर्मली जितने भी सेल्स हैं हमारी बॉडी में, हर एक सेल में कितने क्रोमोसोम्स हैं? देयर आर 46 क्रोमोसोम्स, दैट इज 23 पेयर्स. बट सम सेल्स आर हैप्लॉइड सेल्स आल्सो. अब कौन से सेल्स हैप्लॉइड होते हैं? जिनमें सिर्फ सिंगल सेट ऑफ क्रोमोसोम्स, दैट इज ओनली 23 क्रोमोसोम्स आर प्रेजेंट. आई टोल्ड यू दैट देयर आर टू टाइप्स ऑफ सेल्स इन ह्यूमन बॉडी, somatic cells and germ cells. So somatic cells are all the cells of the body leaving the reproductive cells. Except the reproductive cells, all the cells of the body which have the ability to divide themselves to become double. So if one parent cell is dividing into two daughter cells, so इसमें क्या होगा? Diploid cells. So diploid cells में क्या होता है? The cell having paired chromosomes, that is पूरे के पूरे 46 chromosomes जिन cells में होंगे उन cells को हम diploid cells बोलते हैं. Okay, diploid cells and haploid cells. I hope it is clear. Haploid cells have only 23 chromosomes. ठीक है? And diploid cells में 46 chromosomes होते हैं. And you know that normally in a cell, एक सेल में न्यूक्लियस के अंदर 46 क्रोमोसोम्स होते हैं. इन ह्यूमन डीएनए यू कैन से. सो हैप्लॉइड सेल, नाउ डिफरेंस क्या है? डिफरेंस इनमें यही है कि आई टोल्ड यू टू टाइप्स ऑफ सेल्स. सोमैटिक सेल्स जो होते हैं उनमें हमेशा 46 क्रोमोसोम्स ही होते हैं. मींस सोमैटिक सेल्स दे अंडरगो माइटोसिस. माइटोसिस एंड मेयोसिस, आई होप यू नो. माइटोसिस में क्या होता है? जो पेरेंट सेल का नंबर ऑफ क्रोमोसोम्स है दैट विल रिमेन एग्जैक्ट इन द डॉटर सेल्स. अगर पेरेंट सेल डिवाइड हो रहा है टू डॉटर सेल्स में, सो पेरेंट सेल में 46 क्रोमोसोम्स थे, जो दो न्यू डॉटर सेल बने हैं उनमें भी each one of the daughter cell will have 46 and 46 chromosomes. Clear? This is the condition of somatic cell. सोमैटिक सेल में सो डिप्लॉयड सेल्स होते हैं. जितने भी सोमैटिक सेल्स होते हैं वो डिप्लॉयड होते हैं. दैट मींस ऑल हैव, जितने भी आगे डॉटर सेल्स बनते जाएंगे उनमें 46 क्रोमोसोम्स ही होंगे. बट ओनली द जर्म सेल्स, जो जर्म सेल्स होते हैं जो रिप्रोडक्टिव ऑर्गन्स में होते हैं, दैट आर प्रेजेंट इन द गोनाड्स. I'll tell you what are those. So haploid cells होते हैं reproductive organs में germ cells. Germ cells initially they are diploid, but then they form gametes. Germ cells they undergo division, they undergo meiosis. Germ cells में meiosis होती है. Always remember, somatic cells में mitosis होती है. That's why always the new daughter cells which are forming, they have 46 chromosomes in each daughter cell. But in germ cells always undergo meiosis. उनमें meiosis होती है and then they form gamete. germ cell क्या form करता है? gamete form करता है. and germ cell is diploid. germ cell में 46 chromosomes है, but आगे जो gamete produce हो रहा है, जो male gamete produce हो रहा है, उसमें सिर्फ 23 chromosomes होंगे. that means gamete is a haploid cell. Now why is it so? That I will tell. Okay, so for the time now, just take down haploid cell, that means only 23 chromosomes. Diploid cells have 46 chromosomes. Haploid cells, they undergo meiosis. Diploid cells undergo mitosis. Okay, all somatic cells are diploid, and all germ cells after forming gametes they become haploid. And they are represented also, they are represented by single n and 2n. If only small single n is written in brackets, that means it is a haploid cell. N stands for 23. And if it is written 2n, 2 and small n, that means it is a diploid cell. So 2 into 23, 46 chromosomes. Now next term is unisexual. So unisexual means organisms which have only one sex in one individual, like we human beings. In human beings में क्या है, male और female are different. So we are unisexual, we can say, because male have only male character and female have only female character. Male will produce only male cell, and female they will only produce female egg cells. They will produce. So that's why it is said that human beings, they are unisexual, and some plants are also there which are unisexual, like papaya. Papaya में male plant and female plant are different. Okay, so only example you see, details में नहीं पढ़ना है. Then bisexual or hermaphrodite. Very carefully look at these terms because these are going to be used. Bisexual or hermaphrodite. Hermaphrodite means each individual has both male and female sex organs. ठीक है, एक ही इंडिविजुअल में दोनों organs होंगे, and they will produce sperms and ova in one body only. Example is majority of flowers. Flowers में कैसे reproduction होती है, sexual reproduction, we will study after this. And earthworm and garden snail also, starfish also, means they have both the sex organs, male and female, so they can produce, means a new individual, new offspring can be produced from only one parent. Bisexual, which we have studied asexual reproduction में. So in that, no different male and female is required to produce new organisms. Clear? Then next is gonads. What are gonads? Gonads are primary sex organs. So male gonads, male reproductive system, they have testes which produce sperms, and female gonads, they are called ovaries which produce eggs. Okay, so these sperms and eggs, they are gametes, you can say. So this is a male gamete, that is sperm, and female gamete is egg. So fusion of sperm and egg, that process is called fertilization. Okay, we will study in details. Then somatic cell and germ cell, I told you. What is somatic cell? Any cell of a living organism other than reproductive cells, that is called somatic cell. And germ cell is a reproductive cell that gives rise to the gametes of an organism. I told you that germ cell produces gametes. Now, two more terms are there: external fertilization and internal fertilization. External, from the name only आपको पता लग जाएगा, external means outside. So fertilization that takes place outside the body of the organism. इसमें क्या होता है? Both it happens mostly in some fishes, some aquatic animals में ये process होती है. Both male and female discharge their gametes outside their body, and fertilization occurs outside in the environment. So this is the method in some aquatic animals, in some fishes it is there, but it is not a sure shot method of reproduction, because we don't know that if they will meet or not. So female and male, they are discharging their gametes outside their body, so that both gametes will fuse together or not, we don't know that there are chances or not. So this is not a sure shot method of fertilization. Internal fertilization means, in this, male discharges the gametes into the reproductive tract of the female, and it occurs inside the body of the female organism. It takes place in human beings and animals also. Okay, so these are the terms which you should know. Again, I will share the slide, you can go through it again. Now why this sexual mode of reproduction is required? Because we have studied so many methods, हमने asexual reproduction के पढ़ लिए हैं, that organism can be grown by asexual methods. Then what is the need of having this sexual mode of reproduction? Because as I have told you that what is happening in the reproduction, सबसे पहली process क्या हो रही है? That is DNA copying. डीएनए कॉपिंग हो रही है. अब डीएनए कॉपिंग, इफ यू टॉक अबाउट एसेक्सुअल रिप्रोडक्शन में आप डीएनए कॉपिंग की बात करो, आई टोल्ड यू दैट दिस प्रोसेस ऑफ डीएनए कॉपिंग इज नॉट ऑलवेज एग्जैक्ट, इट इज नॉट ऑलवेज करेक्ट. सो देयर आर सम एरर्स व्हाइल डीएनए कॉपिंग. कुछ एरर्स हो जाते हैं, और वो एरर्स व्हाट दे लीड टू वेरिएशंस. अब अगर एसेक्सुअल रिप्रोडक्शन में डीएनए कॉपी में वेरिएशंस आ रहे हैं, सो यू कैन इमेजिन सेक्सुअल रिप्रोडक्शन में इसमें क्या हो रहा है? मेल एंड फीमेल इज डिफरेंट, सो टू पेरेंट्स आर इन्वॉल्व. इसमें 2 parents are involved, so each one has a chance of variation, so we can have more and more variations. So sexual mode of reproduction का सबसे बड़ा benefit यही है that it allows more and more variations because it involves 2 parents. अब दोनों पेरेंट्स के जितने दोनों के डीएनए कॉपी हुए हैं, तो डीएनए कॉपी में कुछ वेरिएशंस आई हैं, कुछ एरर्स आए हैं. नाउ दैट एरर्स while after fertilization it will come into the zygote, into the new baby, so it can lead to more and more variations. Okay, then diversity of characters can be there. You know that we all are not same. All the human beings in this world, they are not same, they are different, because so you can imagine that so much of variation is there in human beings because of this sexual mode of reproduction. Then new combination of variants. So in your book on page 133, why the sexual mode of reproduction, in the very first paragraph they have told the importance of variation. इन्होंने क्या बताया है that the creation of 2 new cells from one involves copying of the DNA, as I told you कि क्या कैसे होता है, सबसे पहले DNA copy होता है. Then DNA copying mechanism cannot be absolutely accurate. I told you it is not accurate. There can be errors and these errors can lead to variation. So you can say that it would therefore make sense if organisms came up with reproductive modes that allowed more and more variation to be generated. So अगर हमें asexual reproduction में एक parent से variation मिल रही है, so you can imagine that sexual reproduction में when 2 parents are involved, so we can have more and more variations. Then second paragraph mein they are saying that we can have new combinations of variants. How? They say that I told you DNA copying mechanism is not absolutely accurate, but what happened is that it is not a very fast process. अब हम जो हैं we resemble our parents, हम अपने पेरेंट्स के जैसे दिखते हैं. अब ये तो नहीं कि बिल्कुल ही डिफरेंट दिखते हैं और something any new character is coming in us, so that is not there. But every time जब DNA copy कर रहा है, so some errors are there which are not always expressed in the new offspring. जरूरी नहीं है कि अगर आपकी जीन में कोई डिफेक्ट आ गया है so that will be expressed or not. ये सब पढ़ेंगे हम नेक्स्ट चैप्टर में हेरिडिटी एंड एवोल्यूशन में कि कैसे जीन एक्सप्रेशंस होती हैं, कि अगर कोई जीन न्यू कोई डिफेक्ट आ गया है so will it be expressed or not, कि अगर कोई चेंज आया है, कोई वेरिएशन आई है, तो वो आगे न्यू जनरेशन में एक्सप्रेस होगी कि नहीं होगी. So what are the factors? So that we will study in the next chapter. So here they are saying that this is a very slow process. जो variation की process होती है, जो new generations में variations आती हैं, that is a very slow process because all that gene expressions are involved in them. कि जो genes में changes आए हैं, it will be expressed or not. But it can be done to speed up the process. That's why this sexual reproduction method takes place, because 2 organisms are there. अब जैसे for example my grandparents you take, अब ग्रैंडपेरेंट्स के भी जीन्स में कुछ वेरिएशंस आए होंगे, उनके जो फादर थे उनका जब डीएनए कॉपी हुआ है कुछ वेरिएशंस आए होंगे, उनमें से फिर फादर में जब डीएनए कॉपी हुआ उसमें भी कुछ वेरिएशंस होंगे. देन आई केम, सो देयर आर सम वेरिएशंस इन मी आल्सो, सो व्हेन आई विल रिप्रोड्यूस, सो देन देयर विल बी वेरिएशंस इन माय नेक्स्ट जनरेशन आल्सो. So it comes means, but they will be expressed or not, that is different method. वो कुछ उसमें recessive genes और dominative genes होती हैं, that we will discuss later. So here they are saying that when 2 individuals are involved, so there are more chances of variations, and every time the combination is new. Every time the combination is new. See, my genes are little bit different from my parents, so when my next generation will come, then they will also have some new genes. So every time this combination is new, it is written here, see on page 133, last 2 lines, each combination would be novel. Novel means new. So every time when new combination will come, that will be new, since it would involve 2 different individuals. 2 different individuals involved होंगे, so that's why every time the combination of the genes will be new, and the sexual mode of reproduction incorporates such a process of combining DNA from 2 different individuals during reproduction. But अब ये बोल रहे हैं कि देयर इज सम प्रॉब्लम है, तो इसमें एक प्रॉब्लम भी है. व्हाट इज द प्रॉब्लम? सो दे आर सेइंग बट दिस क्रिएट्स अ मेजर डिफिकल्टी. इट इज देयर इन द नेक्स्ट पैराग्राफ ऑन पेज 134. पेज 134 में टॉप में फर्स्ट पैराग्राफ देखो. बट दिस क्रिएट्स अ मेजर डिफिकल्टी. अगर ऐसा ही है कि डीएनए कॉपी होता जा रहा है, डीएनए कॉपिंग हो रही है, फॉर एग्जांपल देयर इज 46 क्रोमोसोम्स इन द पेरेंट्स डीएनए, सो सेम विल हैपन. 46 से डबल हो जाएंगे. अगर सी आई हैव 46, वन मेल हैज 46 क्रोमोसोम्स, फीमेल हैज 46 क्रोमोसोम्स, सो दोनों की जब फ्यूजन होगी गेमीट्स की, व्हाट विल हैपन? 46 प्लस 46 कितने ज्यादा हो जाएंगे? So then again their generation, next generation, so it is very difficult. Means what this creates a major difficulty. So what is the difficulty इन्होंने next paragraph में यही बोला है that combination of DNA copies from two pre-existing individuals will end up having twice the amount of DNA. So here don't think I have told you about the germ cells, but just remove that from your memory. I am only telling you कि cells में mitosis होती है, उनका DNA content जो है double हो जाता है. Now see, for example, if we have taken two cells, two diploid cells, that means 2n and 2n, so they will produce, these two are for example germ cells, and they will produce gametes of the same 2n and 2n. Here no meiosis is taking place. So they will produce 46 chromosomes. It will female is also producing 46 chromosomes. Then they both will fuse together. Fertilization takes place. Then it will become. You can imagine that दोनों के combined कितने आ जाएंगे? 46 plus 46. How much it will result? Again, these two, this is a male, so it will combine with another female, so they will produce gametes. So then, so agar meiosis nahi hai, so then the DNA content will begin, it will be on doubling. So what will happen? It will mess up the control of the cellular apparatus by the DNA. अगर meiosis नहीं होगी, so what will happen? DNA content double होता जाएगा, and if each generation doubles its DNA, अगर हर generation में DNA double होता जा रहा है, a situation will arise when the amount of DNA will not be accommodated inside the cell. कि इतना ज्यादा DNA हो जाएगा कि सेल के अंदर जगह ही नहीं बचेगी वो DNA को accommodate करने के लिए, and there will be no other cellular apparatus. बाकी जितने भी cell organelles होते हैं उनके लिए जगह ही नहीं बचेगी, and there will be so much of DNA, and a time will come that only DNA